I was asked to talk about livestock gene editing for service. And what I'm going to tell you about is perspectives that I've gained in wearing multiple hats in trying to spin off a startup company in the biotechnology space out of academic discoveries. And so I wear multiple hats as an associate dean for research in a college of veterinary medicine at a land grant university in the United States, and also as the founder of a university spinoff company. So the reason we did this was to try to integrate and streamline the pipeline for advancing scientific discovery into societal impact, going all the way from basic discovery through biological proof of concept, commercial proof of concept, interfacing with the livestock producer, and then eventually trying to get technologies into the food supply. And so at universities, we often operate in this space with the basic discovery and then doing some initial biological proof of concept, but then advancing into the commercial side and interfacing with livestock producers and eventually trying to get things into the food supply is where commercial channels need to be developed. And so we were seeing a need to try to integrate the process to seamlessly transition from a university discovery into commercial channels, especially if they were discoveries that may not have had an immediate interest from large commercial players. And so we've developed some technology that we can apply across all of these livestock species. And so we think about this winding path that we have going from basic discovery to finally hitting impact. And oftentimes as an academic, I didn't think about what were all the different winds and turns that would need to be addressed. And so we started thinking about this and trying to address each. And one of the first things is, after a basic discovery, really getting a sense of whether that discovery is addressing a societal issue or an end user need or interest. In this case, it'd be a livestock producer. And if the answer to that is yes, then we need to think about, what does the commercial channel look like? Is there a market for it? And then, do we have consumer acceptance and buy-in for this? And then lastly, can we gain approval through a government regulatory process to eventually have impact on society? And so we've been interested in navigating this winding path, because I don't have to spend time here because we all understand this, that we have a global food production challenge with decreasing agricultural land, but increasing demand for food. And the demand for animal source protein continues to rise. And so we all understand that one of the solutions here is try to make livestock production more efficient. And so our guiding principle, I'm a reproductive biologist by training, is that in order to achieve genetic gain by improving traits and feeding people more efficiently, we have to drive the propagation of trait driving genetics through germ cells, because they link generations. And these can be genetics in the germ cells that are either naturally formed or engineered. And so if we can achieve widespread dissemination of desirable gametes or embryos from elite or desirable breeding stock, then we can impact genetic gain in a significant way. Our overarching goal is to focus on sperm. We're interested in devising technologies that enable access containing trait driving genetics, and that those can be delivered by natural mating without having to use assisted reproductive technologies. And so for many years, we worked on developing a technology that scientifically we referred to as surrogate sires, but commercially we're now referring to as next-gen sires. And so we've developed this in a range of livestock species, but I'm going to use cattle to demonstrate the concept. And so we've talked about already in this meeting about how breeds like Angus cattle, like we have in North America, have been developed for their carcass characteristics, for the palatability, tenderness, marbling, ribeye area, all of these things. But has also been discussed, the majority of beef cattle production, at least exports around the world, happens in South America, and the Lori cattle are a primary breed. And so these animals have not been developed quite like the Angus has for carcass characteristics, but they're environmentally adapted to these regions of the world. So there's interest in trying to flow Angus genetics into Nelore populations to make hybrids that can be resilient to the environment, but also have improved carcass characteristics. And so we've heard, and I made this slide 3 years ago when the amount of AI that was going on was around 12% in Brazil, and now it's surprising to hear it's in the 20%. So that's great, but still that's only about a quarter of animals are bred by artificial insemination. So even if you can take cryopreserved sperm from an Angus sire, use artificial insemination in the Lori breeds, it's still only around a quarter of females that are being bred. And so we've devised an approach where we try to isolate the sperm producing stem cells out of testicular tissue of a donor male. In this case, it would be an Angus. Put those cells in a culture dish and try to expand their numbers, and then transfer those into the testes of recipient bulls, such as these Nelore recipients that would make Angus sperm, could breed by natural mating and have hybrid progeny. We can also integrate direct germline editing into these cultured cells, as Simon Lilick already talked about. By being able to put them in a culture dish, you open a window of opportunity to do direct germline editing, and therefore could transfer germ cells that have trait driving genetics brought about by something like CRISPR-Cas. And so there's some key aspects in developing this. One is the ability to achieve in vitro expansion of the donor sperm producing stem cells. The other is to try to create recipients that are germline ablated, so that the only sperm they'll make are the sperm from the transplanted donor stem cells. And so to show you, we've worked on the spermatogonial or stem cell culture aspect in livestock. It stems off of work that I did as a postdoc many years ago now in developing culture conditions for mice. This is what mouse sperm producing stem cells look like in a culture dish. We have a proof of concept pipeline where we can show they express the biomarkers they should express. They can exponentially expand in the culture dish. And then the real key is that they can make sperm when you take them out of the culture dish, put them back into testicular tissue. And so we've worked on this in the pig as well, and we can now check the boxes in our proof of concept pipeline on the pig. In cattle, we can check almost all of these boxes. In development right now is testing the ability for them to make sperm at transplant, and we've also done this with sheep. And so having tackled the in vitro expansion of donor sperm producing stem cells, we're interested in, can we make recipients that are ablated of their own germline, but their testes can still harbor sperm production when we transplant stem cells. And so the approach we took was to use genome editing of the Nanos2 gene. And we chose the Nanos2 gene because we knew from classic studies in the 90s using lower order organisms like the fruit fly that have a single Nanos gene. If it's inactivated, then it's both a male and female sterility, but the flies are viable. In mammals, there's 3 genes, including the mouse, three Nanos genes, Nanos 1, 2, and 3. Some embryonic stem cell gene targeting studies showed that if you knock out Nanos 1, there's no phenotype. Both males and females are fertile. If you knock out Nanos 3, it's a male and female sterility problem because primordial germ cells don't survive. But knockout of Nanos 2 produced a male-specific sterility. And so several years ago, we started working on different species, knocking out the Nanos2 gene using CRISPR-Cas9 gene editing, and we're able to show in cattle, pigs, replicated the phenotype in mice, but also in small ruminants, that when we knock out the Nanos2 gene, it's male-specific sterility. And so I'll just show you one of the results with cattle. And so in my lab, we primarily use electroporation to introduce CRISPR RNPs. In the case of the Nanos2 gene, it's a very simple gene to edit because it's a single exon, all around 400 base pairs in every mammalian species. So we can design a double guide RNA approach to get really specific, introduce by electroporation the CRISPR reagents into a zygote, transfer the zygote, and directly off of the embryo, we can generate at a pretty high efficiency animals that have biallelic editing that takes out a major portion of that coding sequence for the Nanos2 gene. And so in this bull calf right here, he had 160 base pair and a 510 base pair deletion alleles. And when we take a biopsy of his testis at 4 months of age and stain for a biomarker of germ cells, you can see in the wild type, the brown stained cells within these tubules are germ cells, but there is no germ cells present in the Nanos 2 edited bull. And so we can take this bull and we can transplant donor sperm producing stem cells. In this case, we took from a wild type donor, a Holstein bull calf, put in this guy has Angus base genetics, and take him out to adulthood and look to see if he makes sperm. And what we can find is that beginning about 180 days post-transplantation and persisting even to today, we can find an abundance of sperm in the ejaculate that is reaching what would be a normal wild type bull at the same age level and normal motility. We can use in vitro fertilization to create embryos, genotype those embryos, and show that the embryos possess the genetics of the donor and not the recipient, so it proves donor-derived spermatogenesis. We can also take these bulls and we can subject them to natural mating, and this is just an ultrasound picture to show that pregnancy can be achieved by natural mating with a Nanos 2 knockout bull that's been transplanted with donor sperm-producing stem cells. And so we think we're at this point, and I just showed you the cattle work, but we've done similar work in the pig, haven't taken it quite as far to achieve high levels of donor sperm production in the pig or in the small ruminants. But what we know is that we can get donor sperm production. We're at the point of refining that to be able to be commercially relevant. But we think we're at the point in cattle where we are entering into this commercial proof of concept. And so we've started to think that we can now have a custom genome editing for livestock producers, both in this next gen sires breeding tool, but also in creating germline edited traits to produce offspring with producer traits that are of producer interest. And so I've started to engage with livestock producers in the Pacific Northwest of the United States to figure out what are the traits that they're most interested in improving with something like genome editing, and they fall into these 4 boxes. And when it comes to next gen sires, in talking with beef cattle, sheep, and goat producers, they're primarily interested in amplifying the breeding of desirable or elite sires, increasing the access to new breeds, and preserving the genetics of legacy breeds. When it comes to germline edited traits, beef cattle producers in the Pacific Northwest, United States are mostly interested in dehorning, getting resistance to bovine respiratory disease and East Coast fever, increasing feed conversion ratio. And I keep telling them that's a really tough one to crack. I don't know if we'll ever get there, but they're also interested in trying to increase polyovulation rate to get twinning. In sheep producers, they're also interested in the feed conversion ratio, increasing it. They're interested in scabby mouth resistance, which is a pox virus, so could be a disease resistance trait. They're also interested in genetic tail docking. And so I think these are things that we can get after potentially with germline editing. And so I thought that I would finish up in my second to last slide by talking about the experiences that we've had so far in trying to navigate the US federal regulatory landscape for some of these technologies. One of the questions we've been faced with is who is responsible for gaining federal approval? We as the developer or the end user? As the developer, we're talking about the university, we're talking about the startup, we're also talking about potentially sub-licensees of the technology. Who's responsible for obtaining that federal approval? Or is it the end user? Because the end user is the one that would be putting these animals into the food chain. And so I think that's a question we're still trying to figure out. When it comes to the Nanos 2 edited animal, one of the questions that always comes up is it a welfare concern? Well, in the case of Nanos 2, the only tissue that the Nanos 2 gene is expressed in is testicular tissue. The only cells in the testicular tissue are germ cells. And so a knockout of Nanos 2, I always like to say that those males don't even know they're sterile. They still produce lots of testosterone. They'll mate just like anything. Does it alter the composition of the edible product? Again, is germline restricted? So I guess testes could be an edible product. But again, it's the germ cells that express Nanos 2. So if we knock it out in that animal, those germ cells die. They're not even present. And then lastly, what is the environmental impact? If these animals were to get into the wild and intermingle with wild populations of cattle, well, this would be self-regulating because if that is propagated, the males are sterile. But what about the offspring of a next-gen sire? Well, if it's coming from non-edited donor sperm-producing stem cells, those offspring won't possess a genome edit. So is this going to be regulated or not? If it's trait edited donor sperm producing stem cells, the genome alterations, if they could be attained with selective breeding, will this be regulated or not? So these are all things as a startup company in this space is trying to navigate. And then I want to finish with this, and I'm channeling Perry Hackett here because 20 years ago he wrote an article that said genetic engineering, what are we fearing? And he introduced me to that article a few days ago. And so I decided I'd try to end in that. And I'm going to consider genetic engineering as gene editing. And I'm not going to be here for the discussions on what triggers a GMO. So I wanted to end with my thoughts on what I think is a GMO in livestock. So I think these DNA changes that we can get from CRISPR-Cas generated indel mutations or polymorphisms, we need to be thinking about, are these different, really different than changes from endogenous double-strand break repair, replication errors that happen, or meiotic recombination? When we think about concerns of changing DNA sequence with gene editing, this idea of generation of a novel protein that might be an allergen, well, I'd argue that it's also possible through random mutation that occurs with DNA replication and meiosis. To the question of, will it have a negative impact on welfare of the animal? I think this is self-regulating. As a developer or producer, I think we're unlikely to advance genome edits that would reduce the efficiency or the health of the animal. And so those would just fall out of the pipeline anyway. Thinking about negative environmental impact, I'd say that this is also possible through just random mutagenesis and selective breeding. An altered composition of the food product. I think this is multifaceted. All animals, including non-edited, are impacted by the diet and the environment. The composition of their edible product changes. And so, in my opinion, a GMO trigger should be something that is an incorporation of foreign DNA into the genome that would not be able to arise without human intervention. And in that perspective, animals with genome edits that are changes in DNA that could arise in nature should not be triggered as a GMO. I'm wearing my academic hat with that one. Thank you.